The Move, Look, and Listen podcast with Dr. Doug Stephey is brought to you by Audible. Get a free audiobook download by visiting audibletrial.com/inbound. We'll have a link in the episode notes for your convenience. Many of the books mentioned here in the podcast are available in Audible. Sign up for a free 30-day membership trial, and you can download any book you like. Support the Move, Look, and Listen podcast with Dr. Doug Stephey by visiting audibletrial.com/inbound.
If our two eyes are not working together well as a fast, synchronized team, our internal map quest continues to be off. It's consistently inconsistent with our ability to judge time and space. Those that don't feel well grounded, those that have some measure of anxiety, oftentimes it starts in the visual system. If you can't move, look, and listen in a fast, accurate, effortless, sustainable, age-appropriate, meaningful way, you're in a world of hurt. There's a whole world in vision and how it affects brain function that no one's ever shared with you. 2020 is perceived as the holy grail of going to the eye doctor. Well, I'm here to change that paradigm. Hello, and welcome back to the Move, Look, and Listen podcast. We are going to talk about everything that is magnocellular, vision-related, and more today. And our guest today is esteemed Dr. John Stein. You don't know him, most likely, but you're going to at the end of this episode. I am so grateful that you've agreed to come and talk about this because, well, the honest truth is that there was an optometrist back in the 1930s, A.M. Skeffington, who wrote this 4-circle Venn diagram of the visual process. And one of the circles he referred to as the circle of centering as a representation of visual attention. And it oftentimes got referred to as the where visual pathway, or as the neuroscience has begun to catch up with things that he knew and wrote about back then, I think we really know now as the magnocellular visual system. But before we launch into the topic, Dr. Stein, would you briefly tell the audience kind of who you are and what you do and what you have done?
Well, I am trained as a medical doctor, and I was training in neurology, but I got interested in research into the visual guidance of movement. And I suppose my main claim to fame is discovering the pathway of what we now call the magnocellular system from the visual cortex to the cerebellum. And the cerebellum is very important for the control of movement and particularly of eye movements. And that's how I got interested in the role of the visual magnocellular system in reading problems. One of the things I did when I was what I call a proper neurologist was I used to run a clinic for children, actually, with neurological problems. And a common problem in children is a tumor afflicting the cerebellum. And I was amazed to find that the children presented very often with a sudden difficulty with learning to read. And when you looked at their eye movements, you found their eyes were flickering all over the place. And they were saying they couldn't see the words and letters because they moved around all the time. And that's what really got me interested in why the cerebellum is important, or the input to the cerebellum is important in reading.
And one tiny comment on that is that when those kids tell us that they could see the words on the page, this had nothing to do with seeing 2020. It was a completely separate event.
Absolutely, yeah.
So having said that, let's just get right to it. And how about if you give us a brief description of what a magnocellular neuron is and how it relates to a parvocellular visual neuron. Can we do that?
Right. Yeah, of course. The magnocellular neurons were first described in the visual system in the retina, because there are really two main kinds of retinal ganglion cells. The retinal ganglion cells are the ones that translate, as it were, light into nervous impulses that then go to the visual cortex. And those ganglion cells fall into two classes, really. Big ones called magnocells and small ones that are called parvocells. And the big ones are, as their name implies, magnocellular, have a large receptive field, which means that they're not very precise spatially. You can't locate small detail with them, but they are extremely sensitive to the timing of things. So they time visual events, and therefore they're extremely important for the control of movement, obviously, because you have to time your movements in relation to visual input. And they're also important for timing your attention, the way your attention moves from, let's say, letter to letter when you're looking at a word. The parvocellular system deals with the opposite end of the spectrum. That is, it has high acuity, deals with color as well, will provide the detail that you need in order to, for instance, to distinguish an A from an O by its little tail, the A. But that detail could not be seen by the magnocellular system, but it can by the parvocellular system. So you need both. You need the parvocellular system for spatial acuity and color, and you need the magnocellular system for timing events.
Fantastic. And could you describe where do magnocellular neurons get their input?
The magnocellular neurons receive input from mainly the so-called red and green cones. That means the cones that are responsive to red or green light. A very small proportion come from the blue cones, but we only have about 10% of blue cones compared with 90% of red and green. So their main input comes from the retinal receptors, the cones.
The cones, not the rods.
There's a small input from the rods, but during daylight vision, that isn't particularly important because the rods are mainly important for very low light levels.
And then also to help further understanding, because I think when we talk about motion processing as a function of the magnocellular system, I think there seems to be some confusion about trying to equate magno with peripheral vision. But it's my understanding that magnocellular neurons are scattered throughout the retina, even around the macula, and maybe concentrated around the macula.
They're even concentrated, that's true. They're more regularly plated, as we say, right across the retina. So the magnocellular system deals with not only the center of vision, which is where the cones are most prominent, but it also deals with the periphery. But that's not to say that periphery is more important or the center for the magnocellular system. It deals with motion across the whole visual field.
You know, when I talk to patients in my own clinic, and at the end of the day, I'm a clinical scientist, not a basic scientist. And one of the simple descriptions I will tell people about when they're contemplating, well, what about this whole motion sensitivity? And I try to put it in that and other visual function in terms of predators versus prey. And I say, well, look, prey animals survive by being highly sensitized to their environment. They're peripherally very aware. They have eyes on either side of their head. They have almost a 360-degree field of view. They are fast, short-term sprinters, and they have camouflage. So that's how they survive in the wild. And predators survive because we have eyes closely spaced in our face. We're supposed to have binocular vision, use our two eyes together as a team. We're also sensitive to motion in our periphery because if a deer is hiding in the brush off to our peripheral vision and they twitch an ear, hopefully our motion sensitivity neurons will notice that movement, and then we'll turn and look and try to see and use our pattern detection abilities to break their camouflage. And anyway, that's kind of the circle of life. And I think it's really intriguing. And I'm just going to list a couple of things, and then we'll move on to some more of the details specifically about what you know about magno and visually guided movement and reading specifically. But would it be reasonable to say that the magnocellular system, and I'm just going to list some topics, so there's only 5 of them, I think, is the magnocellular system involved in aspects of facial processing, threat detection, attention and memory, balance and gait, and reading?
Well, the first of those, facial recognition, it probably has a small part to play in, as it were, directing your attention to a face. But the details of the face, the magnocellular system can't pick up. They're too small. The creases around the corner of your mouth, which tell people whether you're smiling, those are too small for the magnocellular system to pick up. So it plays an important role at the beginning of facial recognition, but not for the recognition itself.
No, I think that's really true, because I was looking at an article the other day that related the magnocellular system to emotional processing. And really, it could be as simple as, is somebody looking at me angry, happy, or sad, and not necessarily the details. That's true.
That's right. It can actually pick up emotions on the face, but not in the sense of picking up the details that I've just been talking about, which you would need to recognize a face, but the general contours of the face, which indicate threat or anger or whatever.
And that was the second on my list, threat detection. So I think yes. And then there was attention and memory.
Well, I mean, the very important feature of the magnocellular system in the direction of attention is that, of course, it spots. First of all, it spots when something comes onto the visual scene, so it will immediately draw your attention to a new event, as it were. But also, it can track the motion of things across your visual field, so it will direct your attention to follow those motions. So it's extremely important for the control of attention. And in fact, if you look at the projections of the magnocellular system beyond the eye and beyond the lateral geniculate nucleus into the cerebral cortex, visual cortex, the projections are to areas that are known to be concerned with the control of attention, thus to the parietal cortex, prefrontal cortex, and cerebellum. That was my part to play.
And then there is balance and gait. And that's always interesting to me, too, because balance and gait is tied up in visual input, inner ear or vestibular input, cerebellar input, and proprioceptive input of being aware of where your body parts are. And all that's got to synchronize with good timing.
Yeah, that's absolutely right. The magnocellular system plays the part of picking up, in this case, for instance, if you're wobbling when you're trying to maintain balance, it'll pick up the movement of the environment, and you use that to help you stop your wobbling. It isn't essential for balance. We all know we can stand upright with our eyes shut, but it is very important for the precision of balance. And in fact, if you have a weak magnocellular system, as we're saying some dyslexics have, you're not so good at balance.
Well, what's really intriguing to me about this exchange is that I frequently have people that I'll have stand on my exam room floor, turn a circle with both eyes open, then stop. And of course, they're going to tell me if they're dizzy or nauseous at all. And I'm going to watch their body sway. And I find it so fascinating that frequently the worst symptoms and the most sway is when they have both eyes open.
Yes, that is a mismatch because their visual magnocellular system is not working as well as it should. And so it's a bit delayed compared with where it should be. So it is actually, if you've got the two eyes open, which is giving you also two different views of where you are in the world and where the bits of the world are in relation to you, if it's all coming in an asynchronous way, it's going to make things even worse.
Yep, I think about right and left eyes as being two global positioning satellites that have got to be in perfect synchronous orbit. And if you've got two different magno systems, right eye and left eye, that are not in perfect synchronous orbit, man, your data input is all over the place.
Yeah, well, we all have it to a certain extent, but if you've got an impaired magnocellular system, as we think many dyslexics have, then that temporal, let's say, asynchrony is going to be worse, and therefore it will cause those symptoms.
All right. So I love the flow of this conversation. So thank you. This has been wonderful so far. We're going to pack a lot of stuff in this time. So let's move on What we're going to focus on in this episode is the role of the magnocellular system, primarily visual, but you will hear later Dr. Stein's going to share with us some components about the cerebellar systems, the motor systems, and the auditory systems, and how they kind of correlate with magnocellular visual neurons. But we're really going to kind of focus a little bit and hammer in on the reading part. So my understanding is, Dr. Stein, that up until maybe the mid to late 50s and going all the way back to the late to mid 1800s, dyslexia was really considered a vision problem, but that changed in the late 50s or so. Could you briefly speak to that?
Yes, the original definition of dyslexia actually was acquired dyslexia by Berlin in 1880-something, in which he found patients who lost the ability to read, but kept all their other faculties, visual faculties, just they had what he called word blindness. And he said the remarkable thing was that their other intelligence was perfectly normal. So that was the origin of the word dyslexia. That was taken up by various English neurologists and doctors, a GP in Eastbourne, I think, who had a patient called Percy, who was perfectly intelligent, he said, in every other way, but just couldn't learn to read. And he thought that he had this word blindness, specific word blindness, selective disability to read with perfectly normal intelligence in other ways. And as I say, it was called word blindness in those days, and it was very much thought of as a visual problem. And it was only as you quite rightly say, in the 50s that the educational psychologists, actually taking the cue from Chomsky about the importance of phonology in language, decided that dyslexia was a phonological problem and was nothing to do with vision. Now, there will be a lot of people saying that dyslexia has nothing to do with vision. Some people even say that visual problems never cause reading difficulties, which I think is complete bullshit. But that is the way in which the majority of educational psychologists think at the moment. I think it makes no sense at all, because obviously vision is important for reading.
It'd be kind of hard to read with your eyes closed. That's
exactly.
Absolutely for sure.
Even they don't think that blind people can read normally.
Well, although interestingly enough, my understanding is, from what I've read some years ago, is that when blind people use Braille, their visual cortex lights up.
That's right. I mean, I think that they should remember that.
So, yes, they're still reading with their visual brain. They're just doing it through their fingertips.
That's right. And in fact, there's dyslexia in blind people. Then their visual cortex doesn't light up. And in fact, their ability to pick up the letters and words is defective, not because there's anything wrong with their fingertips, but because there's something wrong in their, well, I won't say just visual cortex, but in their pathways that normally underlie reading.
So let's really drill down now on why are magnocellular neurons important for reading? I'm just going to let you ramble on for a bit.
All right. Well, I hope it won't be rambling. Magnocellular neurons are very important for reading. Firstly, because they time visual events. And that means they time when you first look at a letter, or we first move from one letter to another, or move to the next word. And that timing event enables you to sequence the letters in the word or the words in a sentence. And that sequencing is what is absolutely important for reading, obviously. And what you find is that children with these kind of reading problems are very often not that bad identifying individual letters. What they're particularly bad at is sequencing the letters in the right order. And that's because this visual magnocellular system is preventing them sequencing them properly and accurately.
And just to elaborate on that, the sequencing accurately, that seems to be a combination of perhaps seeing the image as a gestalt image to start with, and then being able to sequence a number of gestalt images together. And what I'm feebly attempting to say is if you've got this timing deficit in the magnocellular visual system, am I accurate in saying that you may have trouble identifying accurately individual letters that may look similar to other letters?
Well, that isn't so much the problem, as I say. It is the problem of sequencing the letters properly. So it's characteristic of this kind of dyslexic that they will get the order of letters wrong. So instead of seeing, let's say, God as G-O-D, the letters G-O-D, they'll see them or they'll mistake the order that they've seen them as dog, back to front, or was for saw. Those sort of local letter mistakes are very common, and they mean that the sequencing has been inaccurate.
To the degree that that's true, I don't know how you hardly ever develop word reading efficiency skills when you keep perceiving letters and words that are all jumbled up.
Exactly. I mean, it surpasses all belief that people can expect that you can read properly if your eyes and your impression of where letters are is moving around all the time.
And that was a wonderful segue into this, which is, and I think that you would agree with me because pretty sure you just said it in a recent article you wrote in 2019. But if we're confronted with a child who's reported to have difficulties with reading, it makes no sense to me that you would just have that child do more reading. Because what we want to do is we want to get to the root cause of why are you not benefiting from the reading instruction that we are providing you?
Well, I think it's even worse than that because the phonologists, as I call them, would say what you have to do is drum more phonology into them. And they get quite good at phonology, but they still can't see the order of the letters properly.
Yes. And that's just pound foolish to spend that kind of money when we haven't really got to the root cause of why are you struggling so much to start with.
I absolutely agree.
And not only does it appear that magnocellular neurons will prevent you from learning how to read because of the timing events that we're talking about, but if we drum enough of that into you and you get some semblance of our alphabetic writing system and common spellings that happen in the pictures of the pictures of those sounds, basically different letter combinations and the sounds that they represent. But these magnocellular deficits also sound like they can cause reading comprehension problems later on because you still got a timing problem. So even if you can make sense of letter symbols, letter by letter, or morpheme by morpheme, or syllable by syllable, you're still probably going to have difficulties with sequencing phrase by phrase, sentence by sentence, and then read a whole page and then have no idea what you've just read.
That's absolutely right. Another reason for that is you've devoted so much effort, as it were, to working out what the letters are and the words are, that you've not got enough, as it were, memory space or memory energy to remember what it was that the first word was. So by the time you come to the end of the sentence, you've forgotten what the point of the first word was.
One of the analogies that I frequently use is I talk about having $1 worth of dimes. So if you got $1 worth of dimes and that's all the attention you get to spend, well, none of your dollar should be spent on nutrition stuff, none of your dollar on primitive reflexes, none of your dollar on something called a millisecond timing clock, none of your dollar on motion processing or magno, and probably none of your dollar on focusing, tracking, and teaming. Almost all of your dollar should be left over for higher order thinking skills like comprehension.
That's right. And you need to have those dollars left over.
Yes. So I know of two practical clinical ways to measure magnocellular function. There's the program that Dr. Terry Lawton has created over here in the US, and I read about it in 2019. I read about it 20 years ago, but I'm interested in your input on this one. So my understanding is that over in the UK, you've got a motion coherence program, what are called RANDOT kinetic stereograms.
That's the traditional way of measuring motion sensitivity in the visual system, which is why we use it. Terry's is much better adapted to actually training the motion systems. We don't propose that our random dot kinematogram will actually train people. You'd have to do quite a lot more to it. And Terry's done it.
The kinetogram that you're using to measure motion coherence,
Yeah.
is that available over here in the US?
Anybody who wants it can get it from us, but I know a lot of people who've used it. One of the problems is that it is quite difficult for children to do. It's not ideal for picking up this problem in children.
Let's talk about the microsaccades that happen in eye movement control and how it ties up into the magnocellular system a little bit.
Okay. Well, you probably know that your eyes are never totally stationary. In fact, you have to make these microsaccades because otherwise the pigments in your eye bleach and then you wouldn't see anything at all. So you have to have a small amount of eye movement. And your brain, computationally, as it were, is able to compensate for these. In dyslexics, however, or many dyslexics, the eye movements are bigger and therefore they're not so readily compensated for by the system. And that's why they actually see the things tending to move around. But the reason for this is if you've got a weaker magnocellular system, you've got a less strong feedback system bringing the microsaccades back on target, because each time a microsaccade removes you from looking at, let's say, a letter, what normally happens in people without magnocellular problems is that the magnocellular system detects this, what's called retinal slip, because the retina has slipped off looking at the letter, detects that, feeds back an inverse signal of that to bring the letters back on target. Now, if you don't have a good magnocellular system, that negative feedback control is not so fast and it doesn't do a good enough job to keep the system stable.
Okay, so I want to clarify a point. This is going to be helpful for me professionally. So when we're talking about microsaccades being registered and counteracted by an effective magnocellular system, and if the timing is off on that, it would not be unusual to report that letters or words move on the page.
No, it wouldn't.
But more to the point is that could be happening even under one-eyed conditions.
Yes, definitely.
Okay. So, well, this is helpful for me, and I think my colleagues and everybody else is going to listen to this. So you can have perceived movement of letters or words on a page and other, we'll call them optical illusions, under one-eyed conditions, and probably and arguably you're likely to see more of that if you've got both eyes are open, because now you've got, again, two streams that have to synchronize with each other.
Absolutely. And in fact, if you've got both eyes open, you've got the additional problem of controlling your vergence. And when you're reading, your vergence angle has to be quite high. And if your magnocellular system, which is the main thing controlling that vergence, is underactive, then your eyes will tend to drift off and you will get double vision or tendency to double vision.
And if it's confusing enough, I can tell the listening audience this because I've got the tools by which to measure it. Sometimes your brain will keep both eyes on and you'll see double or blurry or all this movement. And sometimes a brain will suppress one eye in order to get rid of that confusion.
Absolutely. And you get suppression, intermittent suppression.
Yes, fantastic. I just want to clarify something for the listening audience. When we're talking about vergence angle, there's two ways to point your eyes in space: convergence and divergence. Right? Convergence, you cross your eyes; divergence, you move them away from each other. But when we look at items up close to read, one of the skills, and there's a whole lot of them, but one of them is that our brain has to cross our two eyes accurately in space.
You've got to converge. When you say cross the two eyes, it's slightly misleading because what you're trying to do is to point at it in an arrowhead at the point that's close to you, the letter that's close to you when you're reading. So it's true the lines of sight are crossing, but they're meant to cross at the point where you're converging your focus. And of course, that's very important because it has a knock-on effect on accommodation. If your eyes are not converging precisely, then your accommodation is out of kilter as well.
Well, and that was going to be my next question, which was, does the magnocellular system have as a direct a role to play in accommodation or in eye focusing as it does in the vergent system?
But it certainly has an important part in the early stage of accommodation. But of course, the later stages, which require a very precise orientation of the two eyes together, must be controlled by the parvo system. So it's the early stages of convergence and accommodation, which the Magno system is most important for, to get your eyes in the right ballpark, and then the parvo system puts them precisely in the right position.
I usually tell people, and I say, you can't figure out what you're looking at if you can't figure out where to look.
Exactly.
When we've been talking about these microsaccades and this movement and the way letters and words might move on the page, can you speak in any way that I can better understand the relationship, if there is one, between the magnocellular and or Vergence ocular motor accommodative control systems versus the pattern glare test that Dr. Wilkins has? Because I've been contemplating this, struggling with it clinically for years, trying to understand if there's a relationship between the two or they're really separable events.
Well, the pattern glare test is, in my view, and you must realize that Arnold and I don't entirely agree on this, the pattern glare test or the sensitivity to particular patterns happens to be the range of spatial frequencies to which the magnocellular system is most sensitive. Okay? So if you've got a weak magnocellular system, then it's likely that that system will be in some sense defective, and that can explain why you get the pattern glare. That's the relationship, because the spatial frequency sensitivity of the magnocellular system is precisely the range which gives people pattern glare.
What's also interesting to me is that when I'm using that pattern glare test, in probably 90% of people that are bothered by it, they're way more bothered by it under two-eyed conditions than when I do it under monocular conditions.
Absolutely, because under those circumstances, that pattern, the stripes, are such that you can't, it's not so easy to, as it were, focus on a particular stripe. And so if you've got the two eyes open, one eye can be, as it were, concentrating on one stripe, and the other eye can be concentrating on a neighboring stripe, but it won't be fusing because the two eyes are still diverged too much. And so you get an additional problem from that.
Let's move on and talk about your experiences with dyslexics, however we want to define that. And that's tough to pin down because I've got a document that probably has 12 different definitions of dyslexia.
Probably this isn't the time to consider it, but I think at the moment the definition of dyslexia is absolutely crazy.
Yes. Okay. That works for me. But let's talk about the experience that you've had, the research that you've done between the outcomes on the magnocellular system and or brain with yellow lenses versus blue lenses.
Right. Well, this is quite controversial, but I'm very happy to talk about it. Let's start with yellow. We found a group of children who very much benefit from viewing scenes, viewing text through yellow filters. And the yellow filters have to be have a particular transmission spectrum, which equates to what is often called unique yellow. The reason for this, we believe, is that unique yellow is the point at which red, green, and yellow inputs are precisely matched, put it that way, to the normal input to the magnocellular system. So if you give the most favorable input to the magnocellular system by providing the children with yellow light, you will increase the ability or facilitate the magnocellular system to work properly. And what we found is that it can have a dramatic effect, not on all dyslexics, but on some, in improving their reading. But first, I should say, it improves their ability to converge, it improves their ability to accommodate, it gets rid of many of the symptoms that we talk about, blur and of diplopia and the things we talked about a moment ago. And as a consequence, we believe, of that, it helps the children to improve their reading. And sometimes this is truly dramatic.
And is that effect typically immediate, or is that after a week or two of wearing the lenses?
This is usually pretty quick. They say now the letters are not moving around, now they're not going double, now they're not blurred.
So yellow lenses is facilitating better functioning magnoneurons.
Yes, directly, as it were, favoring the magnocellular system by providing them with precisely the input they like.
Now, that's different than blue lenses or blue filters.
Absolutely. Blue filters are the opposite of that. Blue filters should do them harm. And in fact, or make them worse. I shouldn't say do them harm in the long term. And in fact, we've done an experiment in which we compared people who benefit from yellow, their response to blue. And it certainly makes things worse for them.
In the 2019 article that you authored, you spoke about the mechanism of why some dyslexics do better with blue lenses, and it's a different route to the brain.
Absolutely. Now, the blue has to be the right wavelength, around 480 nanometers, and that happens to be the maximum or the peak sensitivity of a new kind of cell in the retina, ganglion cell in the retina, called a melanopsin-containing ganglion cell, or intrinsically photosensitive ganglion cell. And these ganglion cells don't play much of a role in conscious vision. What they do is they feed into a nucleus in the hypothalamus called the suprachiasmatic nucleus. And that suprachiasmatic nucleus is the body's clock. You know that you have an internal clock which wakes you up in the morning and puts you to sleep. Now, it runs all sorts of other daily rhythms. But of course, if it didn't have some kind of light input, it would be pretty useless, because it needs to be synchronized to prevailing day length, which varies obviously from summer to winter in most countries anyway. And this is the case that these intrinsically photosensitive ganglion cells supply input about the amount of blue light that is entering the eye, and therefore it wakes you up earlier in summer and later in winter. It is synchronizing your body clock to prevailing day length.
I've got a couple of comments and then a couple of final questions.
Mm-hmm.
One of my comments for the audience is, and we've alluded to this earlier in the episode, there's visual magnoneurons, there's quote unquote auditory magnoneurons, and there's magnoneurons in the motor system and so on. But we're just not going to have time to talk about all of these other systems. But I just want to mention it in passing.
Mm-hmm.
What I do want to speak to, though, is some of the reading that I've done a while ago, and I think maybe your 2019 paper elaborated on this a little bit. There's a role to play in magnocellular neuron function related to omega-3 fatty acids. I think I have that right, don't I?
You certainly do. That's another line altogether. The point here is that omega-3 fatty acids are an essential part of nerve membranes. In fact, they constitute anywhere between 20 and 30% of the fats in nerve membranes. And you'll know that all membranes are made of fats. And they're particularly important in magnocells because they mediate the rapid ionic fluxes that underlie the magnocell's rapid responses in order to time visual inputs.
Are you familiar in any way with a company over here in the U.S. called Interactive Metronome?
I've heard of it. You've moved to another subject.
well, only in this context. And I'm not going to hold anybody to this, but again, I'm a clinical scientist at heart. So I'm contemplating every day, how do I improve my patients' quality of lives? So here's Interactive Metronome in a nutshell. You wear a pair of headphones, you listen to a metronome beat set at 54 beats a minute. You're given a device to wear around one hand or a mat that lays on the floor that you step on. Then we give you 14 different physical activities. And let's say it's clapping the hands together, like playing marching band cymbals. So the instructional set is, I want you to clap your hands together on beat. So you're supposed to anticipate the beat interval and set that trigger off on beat. And this device will measure it down to a single millisecond. So if your timing is off, so there's something called the PFIT model of intelligence that ties into the parietal, frontal, cerebellar lobes and how they synchronize and integrate. And there's talk about attentional control networks and oscillatory behavior and some really interesting things. And we talk about it then generally as a timing deficit or a millisecond timing clock deficit in this auditory motor tool, and probably related to timing problems throughout that system. And I've been struggling for quite a while now about, does it make more sense to do this auditory motor timing clock training before magnocellular visual training? And maybe both of them together would have a really synergistic effect, but...
Can I just backtrack a bit?
Yes, sir.
There are magnocellular equivalents in all the parts of the brain, all the systems of the brain. So we've just talked about visual magnocells, but there are auditory magnocells, there are motor magnocells, and indeed the cerebellum is our magnocellular system. And that means that if we talked a moment ago about omega-3s, that means that omega-3s will have or can have an effect in improving the function of all those systems. So it's not either or, it's both.
Well, and and it made me think of something that I just want to put out there and have you steer me back on track if I'm too far off the mark. But I'm really intrigued by this because let's say that you've got cerebellar timing problems and magnocellular vision timing problems. Mm-hmm. I could treat your magnocellular vision timing problem, but if I haven't fixed your cerebellar timing problem, you're still going to have problematic outcomes. Or vice versa. I could fix your cerebellar timing problems, but if I don't really address your visual magno problems, you're still going to have issues.
It very much depends upon what's the greatest impairment. If you have a generalized magnocellular deficit, then obviously it would be best to start with something that will improve the whole set of magnocellular systems. But if you've got something that's more or less confined to the visual system, then it's sensible to start with the visual system. And that's a clinical decision that you have to decide which is more important. Very often, the dyslexics we see have a whole number of different magnocellular problems. They're terribly poor on timing. They're very clumsy. That's motor magnocellular timing deficits. They can't hear the order of sounds, so they're not very good musicians, etc., etc. And there, we always suggest they eat more fish or take omega-3 supplements. And very often, that's very effective.
Yep. A couple of final comments today. With what we're talking about, magnocellular neurons throughout all of our body systems, and to some degree, vergence and accommodative control. So having a magnocellular problem, I would think, would have a pretty significant effect on handwriting outcomes.
It certainly does. And in fact, you get remarkable improvements in handwriting with omega-3s, for instance.
Oh, that's fantastic. All right. So one final comment or question before we start to wrap up. I've used Dr. Lawton's program in my practice for probably close to 20 years, and it's an integral part of what I do. But what's intriguing to me is that when somebody's job is to look at these right versus leftward moving stripes at low contrast, there's a lot of people that only see movement that goes in the leftward direction. And when I've done this, and again, I've used this program for many years, I just don't know the mechanism, and I'm curious to pitch it past you because I'm grateful to have you as a guest. But when somebody sees mostly singular movement, either all left or all right, about 90% of the time, people see only leftward movement, and maybe about 10% of the time, they only see rightward movement. And I was curious to know, since I have you on here, if you have any idea why that might be true.
Well, that probably means that there is some, let's say, deficiency, well, there's a mismatch between the two cerebral cortices. And what happens in many dyslexics is, of course, that the right hemisphere is relatively stronger in dyslexics than it is in good readers. So what you would expect is that they would see a leftwards movement on the whole, because their right hemisphere is, as it were, less strong than it would be in good readers.
Dr. Stein, I think you'll appreciate this too as a neurologist. So there's a number of things that I've done in the practice clinically that I don't know how these things come to my mind from a reading, patient encounters, but pretty frequently when I'm going to prescribe somebody a color, and it is typically blue and frequently with base end prism, a small amount of base end prism with each other, like a half a diopter of base end prism.
The thing is that if you use base in prisms, you say, because most people have used base out prisms.
Nope. This is base in, and it's either a quarter or a half a diopter in each eye, that no more than that.
Right. That's promoting divergence.
Well, see, but from a strict optical standpoint, that's true. But I think there's an argument to be made. I don't know how to prove this, by the way, so it's theory only, but it's experiential theory. I think it's doing something neurologically, not just optically.
Well, I can see a reason why that might work. I was just going to attack you for base out prisms, but actually base in. Well, I'll attack those who use base out prisms. Because the point is it relieves the version system of having to do the work of keeping the eyes pointing at a near point. So I think that possibly your base in prisms are actually causing divergence, which is encouraging the vergent system to work even more, even if they're very small number of diopters, which that is.
And I'll admit that the most common combination that I'm going to prescribe is a light blue with this low amount of base in prism. And invariably, if I have somebody look up at my fluorescent panel and try to take 2 or 3 deep breaths, and then I show them the prism, the ease at which they breathe is significant, and the volume of which they can breathe is significant. And it's immediately measurable. And then when I take the prism and color away and ask them to breathe again, now their chest expands about 30% of what it did before with the lenses. They feel them not being able to catch their breath. I've looked at kinesthetic sensitivity. I've looked at the palmar reflex. When I touch somebody's palm, yes, it's usually annoying. And the people that I'm going to do that with are likely to be annoyed. So they're annoyed by the light touch when I'm doing it. They're bothered by the after effects of when I stop doing it. They got to rub their hand away to get rid of that message. And then I do it with the color and the prism. And oftentimes it's nothing. It doesn't feel like anything when I'm touching them. And it stops when I stop touching them.
It's the big autonomic effects. Well,
big.
I think it's quite possible. I would say most of that's the blue rather than the prisms.
You're probably right, except for when the combination is the best. And every once in a while, I don't know, I'm going to say 10, 20% of the time, people will prefer the prism and not the color. And every once in a while, they'll prefer a plus a quarter or a plus a half a diopter lens in lieu of the prism or the color, and sometimes low plus and no prism and color. And then I'll tell you this, but I've been taught by a couple of different sources that one way to determine the activity of a moral reflex is to basically do a trust fall. So you have them stand, I get behind them, hands on my shoulder blades, they put their arms out, I tilt them back about 30, 40 degrees, and then I tell them I'm going to do this, but I then drop my hands away about 4 or 5 inches, and then I listen for whether or not they go, or their arms go out, or their foot steps back, or their body crumples to the floor. And I have a lot of these videos. I've got videos of somebody being ridiculously bothered and dysregulated by that trust fall until I put the color and prism on it, and then it almost normalizes like that.
I'm convinced that there's a big tie-up between the autonomic systems and what we call the ordinary central nervous system.
I have surmised, and again, I didn't make this up out of the blue. This is really from a number of patient experiences that I've had over the years. But again, as a neurologist, could you plausibly think of an explanation between magnocellular function and gut function?
Oh, yes.
Glad to know that I'm not off the mark.
I mean, it's indirect, of course. Well, the first thing, of course, is omega-3s, because there's a clear benefit to your gut microbiome of taking omega-3s, just as there is omega-3s benefit your magnocellular systems. That's the sort of most direct connection. But there's also an interesting one which involves the vagus nerve, because the vagus nerve is all the time reporting on what you are eating, on what your gut's doing, about how distended it is and how much air is in it and so on and so forth. In particular, how much glucose, how much protein, how much amino acids and so on. All that's going up the vagus nerve to the nucleus tractus solitarius. And that is, among other places, reporting to the hypothalamus, which is receiving information from aspects, let's put it that way, of the magnocellular system. So there are lots of indirect ties up. So yes, there are.
Oh, and then probably the simplest tie-up is the dysregulated balance of the autonomic nervous system. When somebody says, "Oh my God, you just scared the shit out of me."
Yeah. Well, that phrase, I only realized quite recently that it actually means what it says. You defecate by mistake.
Right. It's overwhelming fear.
Yeah.
I've never had anybody crap or pee themselves, and I've had a couple of people come close, but nobody's ever vomited in my office yet.
Now, well, that's interesting, because actually you're vomiting. Of course, well, as I said earlier, the blue glasses can help people from car sickness, and that's probably because you know that the vomiting center is very close to the vestibular nuclei, and so if you get a mismatch between the inputs from your vestibular nuclei and your eyes, which is what happens if you try and read in a car and you get car sick, what the blue glasses seem to do is adjust that balance, so you're no longer car sick. So I'm surprised you never get sickness, because we do quite often but we deal more with children, I guess. Do you often deal with children?
Oh, yes, absolutely. Yeah, I get a whole bunch of kids that get car sick.
Yeah.
And if I can get a favorable response out of Prism or Color, I will prescribe it in a heartbeat. I give them behavioral surveys to take home. Nobody's ever accused me of never sending them enough articles to read.
Yeah.
So I usually say, here's the questions I want you to take home and answer about your child. Let's try the glasses. You go back and look at the behavioral surveys 2 to 3 or 4 weeks after the glasses. And if there's not a marked difference, then you're going to have to do some kind of vision therapy with me. Could be primitive reflex therapy, could be interactive metronome, could be magno, could be binocular therapy, could be working memory and executive function therapy. But if I don't dramatically change your quality of life with just the glasses, then here's the backup plan, including discussion about nutrition and gut function.
And how often do they have to come back? And how often is the color enough?
I would tell you that with most of the kids that I'm going to assess, it's that group that have either been diagnosed with ADD, ADHD, on the spectrum, thought to have a specific learning disability with no explanation as to why. I would tell you that probably not even 5 or 10% of those people, the glasses are sufficient enough. They need other interventions. If I do it with the adults who manage to survive compulsive schooling, and it's interesting because one of the statistics I read a few years ago was that the majority of people in the U.S., even after graduating college, never read a single book cover to cover.
Yeah, absolutely. Yeah.
So they take themselves out of the game in a lot of cases.
Yeah.
So in that group, I would tell you that probably, I'm going to say 25 to 30 and older, I would probably tell you in that group, 85 to 90, 95% of people are delighted with their quality of life with just my glasses. And we end up not doing therapy.
So, well, We find with our blue glasses, particularly, we usually have to do more things like you. But with the yellow, you can have dramatic changes that last forever, or seem to.
Have you had any experience with the yellow lenses, or the blue for that matter, but the yellow lenses, do they wear the glasses forever, or do they wear them for 6 months, a year or two, and then it's almost like the brain has retrained itself to speed up magno, and then they don't need them anymore?
Particularly with the yellow, that's true. Often with the blue as well, but the blue takes much longer.
There must be something I'm missing about trying to figure out who's the best candidate for yellow, because I don't prescribe it that often. And looking at your body of work, I should use it more often, but I can't figure out who to give it to.
Well, we find that the children who present with mainly vergence problems and blur are helped more with yellow, whereas the ones where the letters are sort of buzzing and moving around, particularly if those who have headaches, blue is better.
Yep.
So the ones with vergence problems, yellow helps. The ones with vergence and accommodation problems, yellow helps. The ones with, as I say, this sort of buzzing of the moving of the letters and words, blue helps.
There was an optometrist that I met 11 years ago who I ended up giving her, I think, color in the low base N. And she said the buzzing in her head went away. And I remember asking her, and I said, "What do you mean the buzzing in your head went away?" And her comment to me was, "Well, if you would have asked me if I had a buzzing in my head, I would have said, 'What are you talking about?'" And I wasn't even aware that it was there until it wasn't there. It's so much fun seeing patients like this and learning from them about what they do and don't experience. Well, Dr. Stein, thank you so much for coming today. This has been a highlight of my year because I've read your work for 20 years or so.
I'm flattered.
I'm telling you, this was when you said yes, I was beside myself. This has been a good week for me. I've had some people this past week or so that I just emailed on the chance that they would say yes, and lo and behold, here you are.
Well, we're all sitting at home, locked up by the.
Well, that might be true.
It's to your advantage.
It's to my advantage. Yes, people that I might not have got to say yes before have said yes now. So, thank you so much for your time. I really appreciate it.
My pleasure.
Thank you for listening to the Move, Look, and Listen podcast with Dr. Doug Stephey. All of the resources mentioned in this podcast and links to learn more about our guest can be found in the episode notes from whichever podcast app you might be listening with. You'll also find a link to Dr. Stephey's website, which is stepheyoptometry.com. That's STEPHEY optometry.com. You can also call the office at 626-332-4510. We'd like to take a quick moment to thank our sponsor, Audible. If you love podcasts, you'll love listening to audiobooks with Audible. Many of the books mentioned here in the podcast can also be found at Audible. Dr. Stephey has a free audiobook waiting for you as a gift just for listening to the Move, Look, and Listen podcast. Simply click the link in the show notes to audibletrial.com/inbound. Sign up for a free 30-day membership trial, and you can download any audiobook you like. And if for any reason and at any time you choose to cancel your membership, you get to keep all of your audiobook downloads. It's risk-free and for free for 30 days. Support the Move, Look, and Listen podcast by visiting audibletrial.com/inbound. We really do appreciate you listening. And until next time, for Dr. Stephey of the Move, Look, and Listen podcast, I'm Tim Edwards with the Inbound Podcasting Network.